Hello, I'm Todd Gillam. Today I'll be presenting on the formation of a particle film on substrate for the detection of flotation frothers via gravimetric detection. My project, Research Project 7, sits within the flotation research projects of the PRIF Mining Consortium and is concerned with developing a sensor for the detection of flotation frothers. Flotation frothers are a class of compounds that are used as surfactants to produce hydrophobically decorated bubbles in a flotation bath. So here we can see that these bubbles rise to the top through a mixture of copper sulfide ore material, and they separate the good quality ore from the lower quality ore through a mediation of a xanthate collector, which decorates the high quality ore particles and hydrophobizes them, allowing them to attach to these bubbles and accumulate at the top of the column. This is then collected and goes through various processes to obtain that copper. To achieve real-time detection of these agents in solution, we require a system called a quartz crystal microbalance. Quartz crystal microbalancers rely on an oscillating crystal that shimmies at a particular frequency and dampens when it interacts with substrates that bind to its surface. Within this project, we're aiming to make complementary materials that are able to bind to the surface and functionalize these QCMs to mediate interactions with the particular frother agents in solution and give a real-time readout as to their concentration. Presently, there's no means to achieve this quickly and in real time in application. Instead, it relies on mineralogists and flotation experts just looking at sort of the quality of the froth from a very objective standpoint, and may not necessarily represent accurately how that concentration needs to be modulated, which is dependent on the incoming quality of ore. So an improvement to the real-time detection and monitoring of these concentrations is vital to achieving a higher efficiency of copper collection, which has a flow-on effect of improved profitability. Key findings to date have been that cyclodextrin materials can be used to form different structures, be they single molecules or polymers, that interact with these key flotation agents and allow us to develop a platform that is able to selectively interact with them in solution. On the left-hand side of this presentation, I have an example of one of these materials, which I've developed as a polymer and used to validate the binding of cyclodextrins for these materials in solution experiments. In the center, I have a thiolated cyclodextrin that's been developed that has the handles on it necessary to attach to the gold quartz crystal microbalance surface, and this will provide a means of giving that real-time sensing of those flotation agents. Initially, when I started this project, the idea of molecular complementarity lended itself to the development of molecularly imprinted polymers. Now, the principle behind molecularly imprinted polymers involves the isolation of a template molecule, taking that template molecule and its unique chemical characteristics and picking complementary functional monomers that are able to interact with the key sites of that template molecule, and then cross-linking them with a polymer backbone, which forms a degree of pockets and pores that are able to bind with that identifiable template molecule, which can then be removed through dissolution into a solvent and then used as a platform for sensing because of this desorption, adsorption, equilibrium property, where the template molecule, which might be your analyte of interest, is able to bind and unbind in a concentration dependent manner. However, the target compounds of interest when considering the flotation frother agent that is provided for investigation in this study, which is the Interfloat F236N product. The four major components that are providing the frothing effect within this mixture are butoxytriglycol, hexanol, pentanol, and cyclohexanol. Now, each of these components are not necessarily distinct enough or rigid enough in their expression of chemical handles that are suitable for coupling with precise monomers for the development of molecularly imprinted polymers. So we decided that this presents a bit of a challenge that limits the applicability of the molecularly imprinted polymer approach to solving this problem, and instead began to consider other chemistries such as host-guest complexation chemistry, which led us to employing cyclodextrins as a host that is capable of guesting or hosting these guest molecules within a hydrophobic cavity. Now, one of the key features of all of these flotation frothing agents is that they are hydrophobic molecules. Now, cyclodextrins come in a variety of sizes that is dependent on how many sugar structures are bound into their ring. So we worked with beta cyclodextrin on the basis that its size complementarity was sufficient that it should bind well with the compounds of interest that we have. So the reason that these have a hydrophobic cavity is that a lot of the hydrogens that are projected off of a certain orientation of these sugars that are arranged in a ring are able to facilitate van der Waals interactions and hydrophobic binding capabilities within that cleft, whereas the rim or the secondary hydroxyls that are projected out from the cyclodextrin structure, shown on the left-hand side of this slide as a blue ring, are the hydrophilic surface. And this is the area that we were looking to synthetically modify to be able to produce the materials necessary to validate these in application. On the right-hand side of the slide, I have an illustration of the concept of this inclusion complexation or host-guest chemistry, where cyclohexanol is able to dock within the hoop structure of beta-cyclodextrin and facilitate this interaction, not only through hydrophobic interactions, but potentially hydrogen bonding with peripheral hydroxyl groups from the cyclodextrin. Now, to validate that this works in principle, I used nuclear magnetic resonance spectroscopy and was able to show that key resonances in these spectra, which represent protons or hydrogen ions that have particular magnetic and chemical environments, or when used in magnetic resonance, they shift and associate with each other between these two molecules in solution, which gives a confirmation that they're binding in solution. So without going too in depth into the ins and outs of NMR, what this means is that I can see the key interactions between cyclohexanol, in this instance, and beta-cyclodextrin, particularly the 3 and 5 protons of these sugar residues, which interact and shift in their environment within the magnetic field, confirming that this has formed a host-guest complex. So to make this utilizable and applicable to analyses that could be conducted to evaluate its interaction with interfloat components, I first had to produce a polymer, so something tangible that's not going to dissolve in water that I can expose to these components and still measure their properties. So to do this, I generated a reactive intermediate of beta-cyclodextrin by converting the hydroxyls of the hydrophilic rim into an epichlorohydrin epoxide. And with this epoxide, I was able to then cross-link these polymers. Then I generated a series of polymers incrementally modifying this approach, eventually using an emulsion polymerization technique, which was able to reduce their particle size, shown through laser diffraction on the left-hand side of the slide and scanning electron microscopy in the center of the slide. You'll notice that they're quite craggy and sort of angular in many aspects. Although small, they did require further grinding to get to this size. Now this was still a prohibitive size for utilization on the quartz crystal microbalance system because they were simply too massive that it was going to be of detriment when using such a sensitive technique. However, they were useful for coupling with other analytical strategies to determine how they interact with the individual components from interflow. So what I'm presenting here is an interaction with the interflow mixture as represented using gas chromatography mass spectrometry. So the left-hand side of this slide shows a chromatogram which identifies the multiple components in the interflow mixture as it comes from the bottle. Now, one thing of note here is there are far more contributing components to the interflow mixture beyond that of what is simply reported as the active constituents on the bottle. Now, taking that into consideration, I used mass spectral analysis to identify many of these components, and I named them and looked at how they were stripped or removed from solution with increasing addition of amount of polymer. And this is what I'm showing on the right hand side of this slide, where as I incrementally increase the amount of polymer sitting in these aqueous solutions of interflow, more and more of these components are being removed, but only those that are hydrophobic and specifically in order of their hydrophobicity. And this is helpful because it does confirm that the cyclodextrins are behaving the way that we want them to. So to overcome the issue of not being able to apply these particles directly to the quartz crystal microbalance due to their size, attempts were made to see if we could reduce that particle size. Sadly, we sort of reached a bottom limit and it was still too high. So back to the synthetic drawing board, I took beta cyclodextrin, I modified it by replacing those hydrophilic rim hydroxyls with bromine substituents, and with those bromine handles in place, bromides can be exchanged quite readily. They're what we call in chemistry a good leaving group. So we're able to take these and synthetically exchange them for another group. And this was done in a two-step reaction with thiourea, and then reduced to the sulfhydryl groups, or SH, shown on the right-hand side of the slide, to provide those thiols that we're looking for as handles for attachment to the gold surface. With this material in hand, we're now at the point where we are functionalizing a QCM sensor. So to date, what I've achieved is binding these components to the gold surface and confirming that that sulfur gold covalent bond has been formed. And I'm beginning to assess how these materials on the surface interact with interflow components in solution through using QCM experimentation. From here, ongoing work will be concerned with characterizing the sensor response to the interflow components and confirming that it works as a viable sensor for real-time detection, simulating a flotation process and replicating its performance in a small-scale flotation operation, which will give an indication as to its scope for upscaling and applicability in industry, and then investigate the scope of the broader applications of these cyclodextrin materials and coatings. This can provide materials that are useful for drug loading and release, or the development of sensors for the detection of hydrophobic drugs, pesticides, or contaminants in food and agriculture. Coupled with the polymers that have been developed, these could also provide sorbent materials that are useful for reclamation of contaminated soils and waters. Lastly, I'd like to give acknowledgement to the project team, my line manager, Associate Professor Anton Blencowe, Professor David Beattie, Professor Bill Skinner, Associate Professor Marta Krasowska, Associate Professor Chris Greet, and research placement students that have joined me over the years and done various aspects of data collection, analysis, and synthesis alongside me and under my supervision. Olivia Mastro-Domenico, Emma Marchison, Ashlyn Austin, Emma Pierce, and Wei Ji. Thank you.